everybody. I'm Manar. Other people in my lab group were Makayla, Rahman, and Sam. The goal of our research, doing research on the reactivation of mutant p53 by small molecules for potential cancer therapy. And just some background on cancer. It's actually the second largest cause of death in the United States. And it's a group of complex diseases caused by damage in DNA, which results in the growth of abnormal cells. The tumor suppressor protein p53 actually protects cells from DNA damage, and it's nicknamed the guardian of the genome. The p53 protein is a tumor suppressor protein which maintains the genomic integrity of the cell and prevents cancer formation, and it's inactivated in most forms of human cancer. Also, in 50% of cancers, this inactivation is due to mutations, making mutant p53 a good therapeutic target.
So the overall goal of the research is to identify small compounds that restore the tumor suppressor capabilities of mutant p53. And the way we've approached this goal is kind of twofold. One, we have developed assays to effectively screen a large compound library. And two, we've been able to characterize the compounds that yield in positive results in the screening assays. So the primary screening method is a GFP-based fluorescence assay, and previous students have developed stable cell lines by transfecting them with a reporter gene vector. If mutant p53 is reactivated and able to bind to DNA, it will lead to the transcription of GFP, which is green fluorescent protein. And this can be visualized under a fluorescent microscope. And for this assay, we either treat cells with 10 or 20 micrograms per mil of a specific compound and observe GFP expression after 48 hours. And the compounds out of about 200 screened that have shown positive results are RD27 and 29, whose structures are shown in the slide.
So we're hypothesizing that these cells are reactivating mutant p53, and this allows the p53 to transcribe the necessary anti-tumor genes and proteins. So one of its roles is that it can cause apoptosis in damaged cells. So we examined whether this was the case in these cell lines. We treated 2 cell lines with the compound. H1299 is a p53 null cell line. It doesn't have any p53 in the cell, and DLD1 is a p53 mutant. So we would expect to only see cell death in DLD1, which has the mutant form of the protein because of its reactivation, and we wouldn't expect to see any change in the null cell line. And here we see there is a difference between the 2 cell lines in terms of cell survival in this assay when they're treated with a reagent to measure the cell death. And there is this small therapeutic window where the mutant cell line is affected, has a lot more cell death than the null cell line. So we want to further characterize these compounds and ensure that they are acting specifically through the p53 pathway.
So we hypothesize that this reactivation of mutant p53 is due to a conformational change from the mutant form to the wild type form. So we investigated this with an immunocytochemistry assay in which cells were treated with the compound and then stained for antibodies for the wild type and mutant forms of p53. So as seen in the figure, there is no binding of the wild type antibody in the untreated cells, but there is binding upon treatment with the compound. So as far as future plans go, we will continue to screen more compounds from our library using the assays that we've talked about today. And then moving forward, we have some future experiments, including an evaluation of p53 regulated genes using a Western blot. So we would look for protein products from those genes. And we also intend on developing an assay to quantify GFP expression.